My name's David Winecove. I'm the CSO of Magnitude Biosciences, and I've been working with the nematode C. elegans for many, many years, and as an academic. But the company we started about 5 years ago, and we work with many companies around the world where we help them solve their problems in product or drug development using the nematode C. elegans. And it's my pleasure to talk to Professor Alex Parker. Um, Alex, would you like to introduce yourself?
Yeah, sure. Thanks for the invitation to speak. So I'm a professor in the Department of Neuroscience at the University of Montreal and a researcher at the CRCHUM here in Montreal. And I've been working with C. elegans my whole life, but more recently we've been studying probiotics as a potential way to treat neurodegenerative diseases.
Alex, and that is exactly why we want to talk to you, because you've done some really amazing work looking at probiotics, and in particular, your recent research showing that a probiotic may be a candidate to help in treating ALS. But can we start at the beginning? Could you tell us how you kind of got into the project of looking for probiotics that might have a therapeutic
Yeah,
use?
sure. So it wasn't on purpose. It was we had been making models using worms for many diseases, but once I started my lab, we started focusing on ALS. And we've been screening, like as you do with these simple systems, you screen for things. We've been screening drugs. And then one day a company called me up. It was Lamlon Health Solutions. And they were wondering, they heard that worms are good for screening, that and our lab did this. Would we be interested in like establishing a partnership to test some of their probiotic strains? And I thought, sure, why not? I mean, we're always looking for partnerships, and it was through a funding mechanism. So most researchers can't say no to that. And at that time, it was they were interested in whether their probiotics were useful in regulating like fat metabolism. They were looking for like a weight loss drug, more or less. And I thought, sure, why not? We can do this. And I hadn't really thought about it. I knew what probiotics were basically from the grocery store. You buy them, they're in yogurts or kombucha, things like this. But I never really thought about it. But in retrospect, it was kind of silly because the way most of us work with C. elegans is that they just eat bacteria that you pick, right? So a probiotic is ostensibly something which is good, has positive effects for generally people. But what about how do they work in a simple system like worms? So we received these strains from Lalmond, and we screened them. We just tested them one by one, and we found that yeah, if you take a look at how the worms accumulate fat as they get older, some of the strains block this or attenuated it, which we thought was cool. And the Lalmond Health Solutions was happy because it's more or less reflected what they saw in their mouse models. But you could do this at a fraction of the cost and like far less time than in mice. And from there, I was like, well, these have huge effects. These probiotics have huge effects on just like fat metabolism in worms. And I was just curious if they had an effect in our disease models. So we tested them. We basically had the resource, we had the tools, we put the two together, and like lo and behold, we found a strain that worked very well, and it was protecting our protecting the nervous system in our ALS worms, and that was it. And we thought, we have this probiotic having this huge effect. What's the mechanism? And we just started taking a look.
So you found a probiotic. You found a probiotic that had a positive effect in your worm model of the motor neuron disease ALS.
That's right.
But one of the questions in probiotics is which strain is good for what? There's lots of different strains out there, and how do
Yes.
you know which strain does what different thing? And so you went through a whole array of these strains, and you found this strain in particular had a positive effect. Is that correct?
That's right. So we had a panel, a list of, well, I can't remember how many strains. It wasn't that many. It was probably, but they've been pre-selected, maybe
Mm.
30, 20. And they're all different. There was a collection of different species. Some of the strains were closely related species, but a bit different. And we found that the one that we just screened for was the one we kept was one that was the most effective, and it was a Lactobacillus rhamnosus HA114. It worked great. There was a closely related species like the R011, but that didn't work, even though they're closely related. So, but it was interesting. But there's, as I said, there's like a huge range of probiotics available that people know, but they all aren't doing the same thing. And this doesn't always become evident. It really depends on how you look at them. Like we developed an assay to look at protecting the nervous system. So this probiotic worked for that. Would this be effective in a different model? We don't know, but that's the point.
But I think one of the things is that many companies are trying to find probiotics that are useful for a particular purpose, but not necessarily have a good way to test them for that purpose. I mean, you could obviously try them in people, but that takes a long time and takes a
No,
lot.
I think that's right, because I think a lot of probiotics are known by the assays or the tests that have already been done. I'm not saying they're limited in number, but they tend to fall in the same categories of like some general human health benefits, which is fine. But I think there's actually a huge resource here of the probiotic strains, which are generally well tolerated by people. Most health agencies and governments have files for these bacteria. So they're ready to be tested, but they have not been put through what we would call specific assays. Like if you have an assay for something like ALS, like we do, then you can screen these things for this application. For something like Alzheimer's disease or behavioral disorders, it really depends on how you use them. I just think it hasn't been done yet. It's a rich resource, but it hasn't been tapped.
No, and so really what c.elegans does is allow you to find specific functions of different probiotics. And you've done that for this particular strain, and you only looked at a relatively small number, and you found one particular strain that was really good in this particular function. It was and closely related strains weren't the same, and that's what we find with bacteria, isn't it? That you can have very closely related bacteria. They might all be lactobacillus something, but they have different functions because they've got different sets of genes and they just do different things.
Yeah, there's like when you look at them, there's enough differences, but these differences count for a lot. And that's just it. It really depends on how you look at them. And I would say that a huge benefit of using something like c.elegans is you can use them to quickly identify. It's like, oh, I found this probiotic. It works great for a model for Parkinson's disease in your hands. But like the next question is like, well, that's great, but what is it doing? Right? Can you back this up? I'm like, yeah, well, with c.elegans, we're really lucky because you have so many resources and tools that are already available, or you can make new ones to figure out what the mechanism is. Because I think one of the keys here is that, I mean, everybody's hoping that the probiotic has some kind of translational value. It can move up preclinical and clinical, but what are you looking for, right? If you just want to see general health effects, that'd be great. But is there any way to track the mechanism? Target engagement of something.
And I think mechanism is really the key to this, because if you want to convince people to take something that's unknown, even though it's completely safe, they really want to know why it works. And that's even more so in the medical community. You know, the medical community is not going to start telling people to take this particular probiotic unless they've got a really clear idea how it works. And I think that's one of the big challenges in the probiotics field, because we do know that some probiotics have beneficial effects. There have been a number of clinical trials in probiotics that show that probiotics have beneficial effects, but without knowing why, it puts a barrier there, right?
Yeah, I agree, because I think there will be skepticism because people will say, well, it's just a general effect. You change the nutrition, that's it. But if you can show that no, it's this pathway is involved, these genes turn on or off, and you can see this multiple times, multiple assays. If it tracks up beyond c.elegans into a more complex system, even better. Because the whole point is, if you can get this to a clinical test, you want to be looking for proof that this mechanism is involved as well. Because without that, I think it's harder. It's much harder to move it forward. Yeah. Even though I think they're a rich source of like protective effects for the nervous system, at least.
And so in the case of HA114, you did find a mechanism using C. elegans. And can
Yeah,
you tell us a little bit more about that?
yeah. So um, we're not sure if it's the complete mechanism, but this is the one that it's important for now. Is that and surprisingly, it involves lipid metabolism. So what we think is happening. Well, let me back up. So the way it worked was like HA114 is protective, and then we looked at a bunch of genes that were being changed, their expression was being changed when you gave the H114 to the worms, and we identified a pathway involved lipid metabolism. And if you start knocking out these genes, the protective effect goes away. So you can make a link. And you're like, oh, okay, well, what is the pathway? Well, it has to do with metabolism in the mitochondria. There's a couple of genes here that are very important. You can track this. And some other effects are changes in lipid metabolism. So you can measure all these things. And so we figured this all out in worms. And we have data from testing this in mice that it's going in the same way. And we're starting a clinical trial right now in Montreal testing H114. And what we'll be looking at are changes in lipid metabolism to prove to hopefully it tracks up from worms to humans. So that's just it. You almost take an unbiased approach by using C. elegans because you're like, if you have a probiotic that's working, you can relatively quickly find out why. What are the pathways that are involved in C. elegans? And then for the most part, these pathways are highly conserved. So you can directly jump to look at vertebrate systems to see if it's there. Because if it's there, that's something you can follow.
So your research showed that there was a pathway where the bacteria provided particular fatty acids that were
Right.
taken up by the worm, used in the mitochondria, which is the part of the cell used to make energy,
Right.
and could through that pathway help the worms overcome this disease situation that you engineered inside these worms.
Yeah.
So I suppose sometimes when we find something in C. elegans, the question is, is that also possibly going to work in a human? Because obviously a worm and a human are different things. But I suppose you looked at the genes, and those genes and those pathways also exist in humans, right? So that gives you
Yeah,
confidence.
that's
Look at
right.
the same pathway in humans.
Yeah, I mean, uh, that is like, would this be a caveat? Yeah, a little bit, because if you find a mechanism that's specific to worms, it's interesting in the terms of biology, but not necessarily for translation. But for the most part, um, it's like these genes and mechanisms that are causing disease are highly conserved, especially metabolic pathways. And that's basically it. We learned two things by using the probiotics and using C. elegans. It's like one, the models for ALS, they show some kind of problem, mitochondrial metabolism. That we don't know why. But the HA114 helps bypass this because basically the ALS worms, they're in an energy deficit, and so they're starving somehow. And this is not helpful to their motor neurons because the motor neurons are trying to fight off the disease. So they're in an energy deficit, which contributes to motor neuron degeneration. HA114 comes along, provides specific fatty acids, and that as an energy source to bypass this. And that's the protective effect, we think.
So it fits already with what we know about ALS. We know that in ALS, there's something to do with energy and the mitochondria. And
Yeah,
so this fits with what you find. And also, I think you found that the lipids alone from the bacteria could
right.
also help the animal. So it seems like the main thing that probiotic is doing is supplying the right kind of lipids.
Yeah, yeah. So that's interesting too, because a lot of people are like, what's going on? So you can do these experiments where you separate out the lipids, the fatty acids versus the proteins, and you test it again, because that's something you can do quickly in worms. It's just like test it again, doesn't take long, and it's like, oh, look, the fatty acids from the HA114 work, but not the proteins. Okay, cool. Which fatty acids? We don't know. So we're trying to take a look. So you can add in additional analysis. We do this with collaborators here, the lipidomics analysis. And so we don't yet know if there's a specific group of fatty acids that are responsible for this, or we don't think it's all of them. But something we can say is so far, adding back individual fatty acids that are enriched is not as effective as the whole bacteria, right? So what we're planning to do next is really map out, is there a specific group of the fatty acids that are important? Which sizes? Does this map to a specific pathway? But we don't know. But on the other hand, it's like some people think perhaps you could just dose specific fatty acids and this will be the drug. But we don't see that. And we think the probiotic is actually a decent source because it's a living organism and it's got what it needs to do it. So we're always talking to people like, what's the best way to go forward? But working with something like worms allows you to get closer to the mechanism to answer these type of questions.
No, that's really interesting because it reiterates that you can do lots of experiments quickly with C. elegans, right? You can try. So you did experiments where you knocked out a particular gene to see if that you thought that's involved in that pathway, and you can see, well, okay, this effect doesn't work without that gene. So you know that works.
Right.
As you said, you can add in different fatty acids. You can see if that works. We can do lots of different things relatively quickly that you couldn't dream of doing all these kind of experiments in a mouse model.
No. I mean, it's just too costly. It takes too long. But so what we try and do is gather the maximum amount of data from the worm experiments to guide selected key experiments in mouse models. It's like, oh, we think it's this mitochondrial lipid pathway. Can we see this in mice? Okay. Do we see motor neurodegeneration in mice? Okay. Does H114 affect it? It protects the nervous system. Okay, that's good, right? But we can't do everything on top of this. So I think the C. elegans experiments really helps guide. It serves as a roadmap for what you should look for in more complex systems. I think it's more than just cost effective. I think it's like a smart way to do it because you really
and
want
also you obviously, no one really wants to do more mouse experiments than they have to, right? Just for the reasons of animal, looking after the animals. So at least it allows you, when you do those experiments in mice, you've got a much better chance that they're the right.
Yeah, and this is what we're seeing. We're in the middle of these mouse experiments right now, and everything's trending. It looks great. I mean, I would say a lot of even for like something as complex as lipid metabolism, we can see it trending in the similar sense as predicted from C. elegans, which is, on one hand, you say that's astounding, but then at the same time, you see this type of stuff a lot with C. elegans. It's often predictive, right? Look at so many genes for nervous system function were identified in worms. A lot of the work from the aging field, these genes, these mechanisms are highly conserved. So I think it's a good way to start.
No, that's really good. And you said that you were embarking upon clinical trials now in people?
Yeah. Yes. So we have funding from the Weston Foundation to do a clinical trial, which is based here in Montreal. A couple other sites in Canada are likely to join, and it's for 100 patients with H114 placebo. But our primary outcome, what we're looking at, is lipid metabolism, right? Because we don't think we have at this point enough, the funding or the patients to get a proper number for the standard ALS metrics. But so what we're looking at is that we're going to be treating the ALS patients with H114 and looking at their lipid profiles before and after to see if it matches, to see what happens. First of all, not much is known about this. And two, if we can arrive at a pattern of what the lipid profile looks like for an ALS patient versus unaffected controls. And if this changes over time with the H114 treatment, this is already indicative that something is happening. The secondary outcomes, of course, will be standard criteria for ALS over time. But that's what we're looking at right now. And this comes from all the work starting in C. elegans, where we see changes in lipid metabolism. We see changes in lipid metabolism, which is corrected in the mouse models by H114. So this is hopefully what we're seeing in the patients as well.
No, it's very exciting. And I really hope it all works out. And I imagine that if you do see those predicted changes in lipid metabolism, it will make it much easier and be able to get the funding to support a proper trial in ALS.
Exactly. So this was our plan, because if we could show that there's something biologically relevant, disease relevant in the patients based on, we'll say, this mechanism, lipid metabolism, and it can be modified back to a closer wild type state with the treatment. This will give us, we think, the data, the information to go bigger. Right.
And when do you think you will know the results of this first?
Oh no, maybe a year.
Yeah.
I mean, I should never say, because
No, it
you know,
always
it's like
takes time.
recruitment. We're happy that the first patient received the treatment last Monday. So we're ecstatic at this point. So hopefully a year from now, we'll have everybody completed, and then they will know. So yeah.
No, that's excellent. It's really great to see the work that you've done. And I really hope that people can see the power of the system and the power of really trying to understand which probiotic does what and the mechanisms that are involved, and that will allow people to develop things much faster and further than they have already.
Well, this is just it. I mean, we're just one lab who did this. There are other labs, other people who have different models. There's a huge collection of probiotic strains, so I'm sure there's more out there. It's just a matter of testing them. So, and that's what I think.
No, absolutely. And I think Magnitude Biosciences, we're trying to increase our capacity all the time. We're working with many different companies on different things, probiotics, but also small molecules and other ingredients, where we try and helping those people develop their products, but also try and understand the mechanisms behind it as well. So
Right.
It's great that the c.elegans community can help much more in actually improving things for people, as well as obviously coming up with some great research. I think the history of c.elegans has shown that c.elegans is a great model to understand fundamental biology, and you can see that 3 Nobel Prizes were won, with lots of different functions and genes have been discovered. But taking it to that level where you actually can use it to help people develop new therapies or new products, I think is kind of, for me, very rewarding, and kind of really shows that it is a worthwhile system, and that we're not, even though the c.elegans community works well together and we all kind of have this kind of shared love of the system, that
Right.
can be useful as well.
Yeah. No, I agree. I think that's true. I think it's a fair assessment. I think c.elegans has a lot to offer in this aspect, and you can just see this from the field. The science is always excellent. It's always innovative, and the researchers often find things that, in a way, that you can't find any other way as fast. So I think there's huge potential there for sure.
No, that's brilliant. Well, thanks very much, Alex, for talking to us. And
oh, you're welcome. That was fun.
Thank you.